And as you'll see, he will claim that rotors are more stable than his movies. But anyway, Omer received all of his education at the University of Tel Aviv in Israel. Actually, the University of Tel Aviv was a pioneer in a concept of a medical physics institute. Early on, they established a whole section of the Department of Physics to be dedicated to medical physics. And a very dear friend of mine, Professor Solange Axelrod, I don't know if any of you know her or heard the name. She was really a pioneer in many concepts like neural control of the heart and alternance, the whole concept of alternance, and quantitating alternance as a measure of heart stability. Unfortunately, she died all too young. But Omer was part of that Institute of Medical Physics and received his Ph.D. and then came to this country for a postdoc in Pepet Khalifa's lab, where he stayed beyond the postdoc and then moved with the entire lab to where they are now, which is the University of Michigan. And he continues to work on dynamics, spiral waves, both in the atria and the ventricles, and some other issues related to cardiac arrhythmias using mapping and mathematical methods. So Omer, thank you for coming.
Thank you, Dr. Moody. I would like also to talk a little bit about how our roads crossed each other. Actually, the medical physics department at the university was mostly focused on medical devices. And Yoram Rudy actually was the one to introduce me to cellular electrophysiology. And I want to thank him for that. I find kind of a pleasure coming here to talk about my work, the work that we are doing in our lab, hopefully to create some discussion here and hopefully to teach you something about what's all about this rotational activity. I had great discussion this afternoon and actually realized that we share a lot of interest, common interest. And some of the things that I'm going to talk about, some of you probably already know, but I still hope that we'll be able to advance this discussion. So what I'm going to talk about is rotational activity, mainly rotational waves in atrial fibrillation. Of course, atrial fibrillation is much more than just the rotational waves. And I'm going to give some overview about the role of those rotational waves, their mechanisms, and how to map those things, those patterns. These are my disclosures. We get support from Medtronic and St. Jude, and I have some company, but none of this is relevant to this talk. So atrial fibrillation, as we should know, it's the most common sustained arrhythmia, and jumping directly to the patterns of waves that are responsible, or possibly responsible for its maintenance. There are been rotors that were observed in patients with atrial fibrillation, but many questions still linger, and the jury is still out there. I listed some of the questions that we have. The first one I would like to mention, it's a more methodological question. What is the effect of the mapping modalities on the observations that you, other people, and we have on that field? After we find those rotors, then we ask ourselves what are the determinants of those rotor dynamics, what controls their initiation, maintenance, and termination. And finally, of course, the important clinical question is what role do they have in maintaining AF? It's important, of course, to realize that these questions are not independent of each other, and mapping, of course, will teach us about mechanisms, and mechanisms will reciprocate and will guide us how best to map those waves. So just to well, this is the outline of my talk. I'm going to start with a background. We'll talk a little bit about the basics of parental activity and rotors, so we're all on the same page. I'll describe some experimental studies on propagation and reentry, talk about ionic determinants of rotor activity, and finalize with clinical studies on reentries. About background, so again, this is touch-to-touch base to make sure that everybody is on the same page. So what is a rotor? I think it's a good way to start. Well, the way I look at it is a special type of reentry, and here in these diagrams I show reentry in general and rotor in particular. When the wave rotates around an anatomical obstacle, an air that is unexcitable rotates, sustained rotation, that's it is reentry. We call that reentry. Other people may call it something else. The idea here is that you have a front that propagates in this diagram clockwise, counterclockwise, and the tail chases that front indefinitely, okay? And the front and tail hit this boundary. Now, a situation could be very different in this diagram. Here we have a very old simulation. Maybe Igor could help me in identifying who did that simulation, because
probably Berenfeld for Hertzov.
Maybe in the 60s even.
Yeah.
I couldn't
No,
find.
not that far.
Not that far?
Not that old. Probably 80s.
Anyway, so in that case, there is a excitation that goes around, counterclockwise around that obstacle in the center, that anatomical obstacle. But as time progresses, as we go here from left to right, we can see that activation detaches from the obstacle, and now the rotation is in the tissue itself, or in the simulated tissue, independently of that obstacle. So we can see that that rotor is not depending on this obstacle, the unexcitable tissue, and we call that core of rotation functional core. And that's, for me and for people that work together with me, this is what constitutes a rotor. The fact that you have an excitable tissue in the center, but it is unexcited. Okay, so that's a functional core, not an anatomical core. So that, in a way, could be a definition of a rotor, the fact that you have no excitation in the center, in the core. However, the parameter of excitation is a little fuzzy, it's a continuous. You can never have very totally uniform tissue. Excitability can vary between totally non-excitable and very excitable. So it seems that we need some refinement of understanding of what rotor is. And here's what I propose to use as that type of critical definition of a rotor. Let's look at these three diagrams and understand what's the difference between this rotor and these two. So if you look at panel A, there is an action potential propagates upward, and that's the isopotential front, and in parallel, the isopotential tail, and they follow parallel to each other, and they don't intersect. The same could be said about this radially propagating wave. The front isopotential doesn't intersect with the isopotential tail. The situation is very different, it's critically different in a rotor. If we follow the front isopotential from the periphery, as we go towards the tip, the curvature grows, and as a result of that, the conduction velocity slows down until the curvature is so steep that conduction velocity fails, conduction fails, and there is no propagation into that dashed area that we designate core. Now, if you wait a little bit, as time passes, that potential will start decreasing, and therefore we're going to have a tail. Now, what distinguishes this pattern from these two is that the front isopotential intersects or meets the tail isopotential. And that is the definition for us of a rotor. That's what makes it special. It's a re-entry. In re-entry, the front and the tail do not meet. In the rotor, they do. Now, how can we have that situation? First of all, this situation is perfectly allowed mathematically. It's one of the solutions of the electrophysiological system. And in that case, we are not describing anything new. However, we have to show that it indeed occurs in the heart. Now, how those rotors can start? Here I have a diagram that illustrates possible scenarios and illustrating how this re-entry, that rotor can start. Let's look at panel A. In that case, we have waves that propagate from top left to the bottom right, and they encounter an obstacle. That obstacle could be either anatomical, an unexcitable tissue, or it could be functional. Maybe that was an ectopic wave that left a refractory area that cannot sustain new action potential. So that front needs to circumnavigate that red front needs to circumnavigate this obstacle. Now, because in this diagram, that obstacle is rounded, and the curvature here at the boundaries of that front is not very high, and therefore those two waves will meet, merge, and propagate towards the right. The situation is different when the obstacle has sharp edges. It could be sharp edges of fibrosis or ischemia, or again, ectopic beat. As we can see here, the curvature at the tip is steeper than this case, and it seems that this wave has some difficulty in meeting. Eventually, in this diagram, they meet and they will propagate together to the bottom right. But if we tweak this condition a little bit and we reduce excitability, then that front has a curvature that is so high that it will now create a detachment from that obstacle, and that detachment from the obstacle will result in pivoting point that is independent of the obstacle itself. And that wave break or that center of rotation can be stable or unstable, but if this sustains once and more rotation, then we would call it a rotor. If you further reduce the excitability, those free ends have difficulty in pivoting, and those waves may eventually go into the boundary and disappear. Now, for today, the objective I set for myself, for us, is to discuss the variable configurations and dynamics of self-sustained rotors in atrial fibrillation in experimental, numerical models, as well as in patients. So I'm going to have, I will cover a lot of ground here, and hopefully I will be clear enough for you to follow. You can ask questions after that. So if I now move to the experimental studies, our main experimental tool is optical mapping. You are very familiar with that technology here as well. We are studying the sheep. We isolate the heart. This is a diagram of the isolated sheep heart. And we can map the atria with up to 3 synchronized CCD cameras. In particular, one of them is connected to an endoscope that can look at the atria from inside, so we don't cut open the atria, and we can look from both sides of the wall, inside and outside, in the intact heart. We use diphornaps, blebistatin to suppress motion, and there are some very technical issues. There is no point in detailing it. You can see here the pictures are seen by the endoscope. This is the posterior left atrium. You can see the veins. And if you look at the left atrial appendage, you can see the thin regions and the thick regions. These are the pectinate bundles. This is a nice picture taken of the endoscope after the experiment. We cut open the atria so you can see how this endoscope looks at the posterior wall. Just a picture. And this is an example of optical recording during atrial fibrillation of the left atrium and right atrium. This is acute atrial fibrillation. This is minimally processed data. You can see in white the excitation. We can see the random activity in the left atrium. It's not 100% stable, but it's one of the most stable ones we ever saw. Now, in the right atrium, the activity is much less specific and difficult to follow. Now, to enhance this characterization of the waves during atrial fibrillation, we apply 2 main methods. One of them will look at the stationary propagation properties, and the other the dynamic propagation properties. To look at the stationary propagation properties, something that is stable for the duration of the analyzed movie, which is about 5 seconds, we use Fourier Transform and extract mostly usually the dominant frequency maps. We could look at regularity as well. For the dynamic propagation properties, we are interested in the presence of wave breaks and rotors, and for that what fits us is the Hilbert Transform that generates the phase maps.
Can I ask a question?
Sure.
When you do the stationary propagation properties, you do the Fourier Transform. Is this where you then take the data and filter it plus or minus some number of hertz around that frequency, and then you use that data for the dynamic propagation?
No.
So these are each independent calculations?
Independent, yes.
Okay.
And the reason we do that, we could talk about that if we have time. The reason I use the Hilbert transform is that that's precisely the reason I use the Hilbert, because it gives me the result for all frequencies. Okay?
Okay.
It doesn't discriminate. If I have to use other methods like return maps or lag methods, then I would be forced to decide about a particular frequency. Even if I don't decide, I unintentionally assume something. We can discuss it later. So this is a demonstration of what is a phase analysis. From my discussions earlier, I realized that some of you are using that already, of course, as many other people. So in that approach, what we do, here is a diagram of the actual potential. In this case, we talk about voltage as a function of either distance or time. And instead of looking at the voltage of fluorescence, which is proportional to the voltage, we color code it to according to the phase of the cycle of the actual potential. For example, in this case, the blue would be the resting, the green the upstroke, and so on. And when we put for each pixel the color of its particular phase, we end up with snapshots that look like that. And in this case, we have a phase singularity. This is the point where all the phases will converge to. Necessarily, by the meaning of the word phase, we're going to have action potentials that rotates around that point. So it's nice that we highlight the center of rotation. And in one snapshot, we already see that the directionality is going to be counterclockwise, because action potential will always move into the repolarized, into resting region. Okay? Yeah, so that's the phase singularity. Mind you, of course, that phase singularity may not last for a long time, and if later we ask ourselves what is a rotor, is a very brief singularity point. Can it describe a rotor? So the answer is, of course, we need to define a minimum amount of rotation to be called rotor, but that seems to be kind of an arbitrary decision. At least one rotation should be present to be called re-entry. So everything I'm going to say later about presence or absence of rotor is whether they last at least one rotation. Now about the stationary properties, here's what we do. Here I show diagrammatically the grid of the pixels from the movies. Each pixel is a time series. We apply Fourier Transform on that time series, get the power spectrum, and take the dominant frequency, the one with the highest power, to be a dominant frequency, and color code it and generate dominant frequency maps. And this is how we combine the two things, the frequency and the phase. And that movie, maybe by some miracle will work, but it didn't work before. No. So the codec is unavailable. So you're going to have to believe me. This was a movie taken from the posterior left atrium and from the left atrial appendage. It shows us singularity points that last for some time, not for the duration of the movie. It was those singularity points moved from the posterior wall to the left atrial appendage and back. So there was a dynamic situation. And here are snapshots of a few times along that movie. So this is the posterior wall, and this is the left atrial appendage. We can see that at the beginning there is re-entry in the posterior left atrium. Here you can see another snapshot with the re-entry, but that re-entry disappeared. And here you see a breakthrough in the posterior left atrium. On the other hand, in the left atrial appendage, at the beginning there was a breakthrough, and then it transformed into re-entry. So that's in the dynamic process. Now the more stationary properties are described here. When the rotor was in the left atrial appendage, the frequency in the left atrial appendage was 8.7, which was the highest that we measured in the atria. You can see that it's not very high compared to the posterior left atrium, only 0.4 hertz. But that small difference was significant, and it was consistent. So the highest frequency is where the rotor is located. Now, later on, when the rotor abandoned the left atrial appendage, the frequency went down to 7.4 hertz. So here, in this example, what I want to highlight is the fact that the highest dominant frequency is often found in sites where we have the rotors. Okay? That's the message I want to send here. Now, I said earlier that we measured activity inside and outside of the atria. I want to describe a little bit of our results here. What I show here are in the top the data from the endocardium at various times during the movie, and this is from the epicardium. This is both on the posterior left atrium. In this case, I show only situations where they are similar. So here we see a breakthrough on both walls near one of the veins. Here reentron activity. Here collision. Here another breakthrough. Here activity sweeping the field of the mapped field. Then here again reentry. Here we see a few singularity points. So sometimes the activity is similar, sometimes it is dissimilar between the endocardium and the epicardium. Now, our methods doesn't allow us good information of what happens inside, but sometimes we have situations which are calling for some speculation that is obvious. In this particular case, if I can have it running, there were two singularity points in the epicardium and endocardium, and although we don't know what happens inside, it makes sense to speculate that there is a line called filament around which that transmural rotation occurs. Okay, we call that scroll wave. Now, not all the time we see those reentron activity on the wall, on the epicardium and endocardium. Oops. Sometimes we see breakthroughs. So this is a case where we saw two breakthroughs, the epicardium and endocardium one. And as I said, we don't know what happened inside, so we could speculate, of course, that there are ectopic activity in the center that give rise to these waves that project toward the surfaces. But there is a possibility that there is reentron activity inside the wall, and the filament itself is hidden from us, but it creates waves that propagates towards the wall. Is that possible? Well, we have a little indication to that in looking at simulations. So we looked at cross sections of the sheep atria. These are very thin wall. This is very thin wall. This is about 3 millimeters thick. And you could ask the question, is that enough to sustain reentry? So the answer is yes. Here is an example, and with a little bit more fibrosis, the reentry is different, but still you could have not necessarily functional reentry, sometimes it's anatomical reentry. Nevertheless, that reentry is possible. That may explain a lot of the data that comes out from Alessi's laboratory and Waldo talking about dissociation between the one side of the atria, the inside and the outside, and this data brings the possibility that indeed that dissociation is a manifestation of intramural reentry. But the data in this is not very strong. This is just simulation. We still don't have the technology to map the thickness of the wall, although some long wavelength dyes may give us deep penetration. Now, the situations could be different on the two walls. At epicardium, we could see reentry, and endocardium, we could see a breakthrough. How do we interpret that? This is again speculation. Maybe we have a filament that goes from the epicardium, and instead of reaching the endocardium, it bends and goes outside of our mapped area, mapped field. Or we could have, here we have two counter-rotating scrollway, or two counter-rotating rotors, or reentries. And on the other side, we have breakthroughs. And that could be explained by a U-shaped filament that doesn't reach the endocardium, but the two ends of that filament is on the same wall. So we have all these possibilities for reentron activity.
Why can't it be reentry on the, or spiral on the epicardium and focal sources on the endocardium?
Yes, possibly. But the first one will dominate. And the ectopies usually don't last long.
It's hard to separate your scenario from that scenario.
Yeah.
Unless you manipulate the system somehow.
Yeah, you could.
Based on the different properties of ectopic focal activity from reentry.
Yeah, you can challenge the atria with drugs to, for example, favor reentry or favor ectopies. For example, increasing IKCH to favor reentry, or overloading calcium to...
Yeah, but it's a possibility.
Yeah. Now, so is there any preferential location for the rotors? So in our hearts, in the paroxysmal acute atrial fibrillation, what we see is that they tend to localize or to cluster in certain regions, those singularity points. And in this case, this is a case of one animal, and these are filaments, of course, speculated filaments at different times in the same movie. They all seem to be around the right inferior pulmonary vein. And if we look at 4 animals and we collect all the singularity points, we are, and we put them on a diagram, of course, this diagram of 4 animals, we see that there is a tendency to cluster around the pulmonary vein. So that brings up the question of what are the determinants, one of the questions that we could ask is what are the determinants of rotor attraction to the pulmonary vein ostia in acute atrial fibrillation. So the answer is, of course, there are many possible factors that can affect that attraction to the pulmonary veins, and here I'm listing some of them. In terms of structure, it could be the wall shape, the wall thickness and the bundles, and the fiber orientations, and fibrosis. And in terms of functional properties, it could be ionic currents and intercellular coupling, to list just a few. Now, okay, yes, so?
You ever see rotors in the right atrium?
In the right, yes, we do. In the ship we see, yeah, we do. Not as often as the left in acute, though.
Does that result in flutter or?
A good question. I don't remember, but not necessarily. It could be. It's likely that they will result in flutter. The reason being because the refractory period of the left atrium is shorter than the right. But I cannot exclude the possibility that they are also atrial fibrillation. Well, anyway, from all this list, I choose to start to focus on ionic currents. Okay, so we will not give a full answer to that question, what are the determinants. Okay, just in terms of ionic currents, but I think it's a good way to start. So let me talk a little bit about ionic determinants of rotor activity. I would like to start by describing 2 old sets of experiments. One experiment is maybe the first experiment in the ventricles that gave us some ionic insight. This is done in a guinea pig, and we are looking at the ventricles now. And this is optical mapping of ventricular fibrillation. And we can see that rotor here. It's the one that is the fastest, is the most stable, and it resides in the anterior part of the left ventricle. Okay? All the rest was slower, and the other singularity points, the other rotors were short-lasting. So we were interested in knowing, is there something special about that ventricle, the left ventricle, that gave rise to that stable rotor? And we focused our studies on IK1 because of various reasons. Its time independency, its role in action potential duration, and excitability. So we isolated cells, and we measured the IK1 in cells from left and right ventricle. And what we found is that the outward component in the left ventricle is larger, significantly larger than the outward component of the right ventricle. When you plug those IV relationship into a computer model, it gives you action potential duration in the left that is shorter than the one in the right, and a little bit more hyperpolarized. That turns out to be important. The tiny hyperpolarization seems to be important. Okay, and then we wanted to see if those action potentials, how did they affect re-entry? So I think these movies don't run, so you'll have to believe me. Yeah, so this one is, that rotor cannot sustain itself. The action potential duration is not brief enough. It doesn't allow the deviation, and the excitability is lower. So that doesn't rotate. And this one doesn't rotate. It should rotate, but it doesn't. Anyway, for some reason this movie does work. What we did here is to plug the left ventricle properties surrounded by right ventricle properties. And here, yes, okay, so this one works. So we were able to generate action potential that is very stable compared to the action potential. Many singularity points around, but they are slower and they depend on the one in the center. Once you stop this one, the other will die. Okay, so the IK1 seem to be important in creating those conditions important for reentrant rotor activity, the acceleration of the rotor and its stabilization. Here's another set of experiments. Here we looked at IKR, another potassium current, delayed potassium current. What we did here is to study that IKR in a dish, and with magnetoflexion we were able to create two types of dishes. One in which the cells are uniformly overexpressed with IKR, and another types of dishes in which there was half of the dish overexpressing the IKR, and the other half we have a baseline IKR yet. Okay? And these are the results in the case of the when you have half and half. So this top half will be base IKR, and the bottom will be infected. So we have overexpression of IKR. So look what happens when this, the rotor is in the basal IKR region. So the action potential duration here is the same as here. So the rotor that is present here doesn't have any problem. Sorry, the IPD here is briefer. So the rotor has no problem propagating downwards. However, if the rotor is in the IKR overexpressed region, situation is very different. Now that rotor is faster than this rotor because the IPD here is briefer, and also there is a little bit of increased excitability. And the IPD here is long, so we cannot have one-to-one propagation upward. And that's fibrillation. Okay? And this is these are snapshots in time-space plot illustrating those results. So when the okay, this one is this is all infected, uniform infection. So the IKR is upregulated everywhere, and the frequency is uniform everywhere. However, when there is a half infected and a half basal IKR, the rotor resides in the infected region, it will be faster than the area that has lower IKR. So we concluded that if fibrillation that is, according to our working hypothesis, the result of rotor plus wave rate, that upregulation of IK1 and IKR causes the acceleration of rotors by reduce the core size and reduce the meandering. So this is the effect of these two currents. I will stick to these currents as well. We studied other currents, but this seems to be the most important one that we found, the one that most affects the rotor stability and frequency of rotation.
May I have a question?
Sure.
So I mean, is that just the two currents produce similar properties of action potential that
Yes.
give you rotor?
Yes.
So presumably, if you have other currents that produce a similar action potential property, no matter what current it is, then you should have a similar
Absolutely
rotors,
correct.
right?
Absolutely correct. Yeah,
The
I don't have
only
I don't have.
The only difference could be because of IK1 effect on the baseline, right?
Yeah, it seems that the IK1. It's a little difficult to conclude something solid from these studies because we don't know how much IKR we overexpressed. There is no way when you do overexpression studies, you don't know exactly how much you overexpressed. So when there is no ground for comparison about how much, let's say, 50% increase in IKR versus 50% increase in IK1, how much they will cause the rotor change. We don't have that unless we do simulations.
Have you done anything with IKS?
Yes, we did.
And
And IKS doesn't affect the frequency of rotors at all. What happens, it prolongs the post-repolarization refractoriness, and it causes a very complex fibrillatory activity. There are many wavelengths in the periphery of the rotor. I can show you tomorrow maybe.
that's quite interesting because the known, one of the first to be discovered, hereditary mutations that cause atrial fibrillation was an IKS mutation.
So that may, it doesn't affect
gain
the-
of function of IKS,
Yeah, well, gain of function would cause...
which is like overexpression.
Yes, yes. So it would, essentially, it increases the post-polarization of fractures. The cell seems to be having more difficulty in recovering, and therefore the wavefront will have difficulty in propagating. There is no, we didn't find, in cell cultures, we didn't find any effect on conduction velocity at certain frequencies that we could base, and frequency of re-entry. These are the results. I'm not sure if that's the same, going to be the same in the tissue. It's supposedly the same, but it has to be demonstrated.
This could be a problem, of limitations with the experiments, because what Dr. Sui said is true, especially when you compare. IK1 is a little different because it affects stability due to its effect on the rest potential. But all three currents will shorten action potential duration if you overexpress them.
Well, we found that in the IKR case, for example, you have also hyperpolarization, but it's transient. So it works during normal pacing, normal propagation. That transient hyperpolarization may not be affecting conduction velocity because it's very brief. However, if you do have a re-entry, and when the front and tail are tied together, then that transient hyperpolarization may be acting like the one in the IK1. The IKS doesn't have that. The IKS didn't show us hyperpolarization.
But I think, I'm just, of course I'm biased, but I think IKS can play a tremendous role. First of all, there is this clinical data and genetic data that IKS mutation that causes. That was actually the earliest paper published on hereditary AF.
I know
from a group in China.
I actually know about the IK1, some mutations in IK1,
Anyway, but IKS is and there is an IKS mutation that is associated with. Let me just finish. There is an IKS mutation that is associated with atrial fibrillation. It's hereditary. It was published from a Chinese family or several families, and shows very clearly that there is a relationship between that mutation and atrial fibrillation. That was IKS. The other thing that is interesting is that beta adrenergic stimulation, which plays a role in atrial fibrillation, is affecting IKS. That's the target, and IKS is increased by a factor of 3 or so, maybe even 5 sometimes, with beta adrenergic tone. That means that then the action potential duration will be further decreased by much. And so I just wondered. I think there is a lot there that could be explored.
But I think that's a finding that was of mutations is that some AFs have very slow frequency. They are very complex, but still slow.
Yeah.
And others are fast and complex.
Yeah.
So the ones that are slow may be having different mechanisms than these two.
There could be another thing here that we're. We all have the same tendency to focus on something that we focus on to the expense of all other things. And when I say IKS is a target for beta adrenergic stimulation, it is, and it can shorten action potential duration, but at the same time, it enhances calcium release from the SR and can cause spontaneous release, which can cause focal activity. So, atrial fibrillation is complicated. It could be both. It could be everything.
But one of the things that maybe that also, you were saying, the beta adrenergic stimulation and also the mutations of IKS. Those manipulations, modulations actually changes the property of IKS. So if your simulation, if you just simply by increase the IKS twice, as large without changing properties of ICAS, you may get a different
Yeah,
result.
no question about it. I mean, yeah, that could be said about everything, yes. We are changing.
I just think we shouldn't lose sight of the possibility of focal activity, because many of those triggering events that would support rotor formation also enhance ectopic activity through the calcium mechanism, and that's another option, another
Yeah.
possibility.
I cannot exclude that. There's no way to exclude that.
It's just I think we shouldn't lose sight of it. That's all.
Now, I would like, regarding the dynamic, the attraction of rotor activity and attraction to the pulmonary vein, I would like to propose that rotor attraction to pulmonary veins is driven by the spatial gradients of transfemoral currents in the LA-PV junction. It's not to say that other factors do not play a role, but the way the ionic gradients are distributed in that junction may affect that attraction. And that's what we were interested in studying. So we looked at data that was collected in dogs in the laboratory of Stenatell, and here we see sections of tissue between the pulmonary vein and the left atrium. And these are the currents that were measured in those isolated cells from those regions. The ITO, the N-type calcium, IKR, IKS, and IK1. In particular, I want to mention here the IKR and IK1, which was earlier in the previous slides shown to have the same effect on rotor activity. However, in the pulmonary, in the dogs at least, it was found that the pulmonary vein IKR is larger than the left atrium, and the IK1 is distributed totally the opposite. So the question becomes now, what is the effect of these opposing gradients of current? And the way we address that is to build a computer model. And here are the models that we built. These are simplistic spatial models that contain an hypothetical distribution of this current. So the current themselves are maximum and minimum of each current is according to the data from Stenatell. However, there's no information about the transitions, so we plotted those, or we assumed those to be sigmoidal type with a space constant corresponding to the distance between the cells that Stenatell measured. And we built three types of models. One was square, the other was basically wrapping that square and creating a cylinder, and then a funnel type of model that is given intrinsic geometry that corresponds to the narrow pulmonary vein versus the wider left atrium. And the first thing we did is to look at the effect of gradient of each different current. So each trajectory here in different color describes the drift of that trajectory of that rotor that was initiated in the middle of the junction, and each color corresponds to different currents. So you can see that each distribution of each current throws that rotor to a different direction. And the next thing to do is to compare them, to put them to pair them. So we now see which one affects where direction. So here are all the currents, and this is the destination of the rotor with each pair. So for example, we can see that, for example, the L-type calcium L-type calcium by itself links to the left atrium, but the L-type calcium versus the IK1 wins and takes it to the pulmonary vein. And you can look at other currents. The important thing is the IK1 and IKR, as I said. So if you compare the IKR and IK1, we see that the IK1 wins because the IKR by itself is taken to the left atrium. So the IK1 seems to, at least in that setting of conductances, wins over the other currents that we measured. And this is just a movie of that same drift. We could put all the currents together now, and we can see that regardless of whether the rotor is clockwise or counterclockwise, when we start it in the middle, it will drift towards the pulmonary vein after few rotations. The question becomes, well, what goes on there? It's again going back to the excitability. How is that drift possible? So we looked at one case in which we started the rotor here in the center, and we drifted towards the pulmonary vein here in this direction. And we look along that red line Now, not on the voltage, but rather on this parameter Hj from the modeling that describes the variability of sodium channels. And here's an inset of what happens during that drift. So the drift is along that line here. This zigzagging is typical of rotor. And you can see that that zigzag shifts towards the pulmonary vein. And in this inset, or this magnification of that region, we can see that the excitability in the LA side is more red compared to the excitability in the pulmonary vein region. And if you look at that scale, we can see that the excitability here is smaller than the excitability here, and the rotor drifts toward lower excitability. It contradicts some things that we may be discussing later, but that's the finding, that the rotor goes to the lower excitability region. Okay? Now, is the geometry affecting that? Well, we studied just one aspect of the geometry, and that's the narrow circle of the pulmonary vein. This is what happens in the vein, in the pulmonary vein junction, in the funnel model, when the tissue was homogeneous. So we started the rotor here. This, the blue is the trajectory of that rotor. It seems to have a more or less steady distance from the pulmonary vein. However, if we introduce heterogeneity in all the kinds that I described before, in that same funnel, we start the rotor here, and it seems to be attracted to the pulmonary vein, and this is illustrated by this graph of distance of the rotor from the pulmonary vein as a function of time. We can see that the homogeneous case is more or less stable, and the heterogeneous case has this attraction towards the pulmonary vein. Okay? So we demonstrated in those simulations that the ionic currents, the landscape of conductances of those currents that we studied lead to attraction to the pulmonary vein, which is consistent with the findings in acute AF. Yes?
So does this ever rotate, just become anatomically reacting?
It could, but not in our case. No, the frequency is higher than the conduction velocity around us. There is some tendency to this one to drift a little bit around. It doesn't go straight. There is some recession if you want. Let's move on. I want the most interesting things. There always no time. How much time do we have?
Just continue.
Okay.
So
Sorry.
anybody who needs to leave can leave.
Okay, so I want to switch gears to go back to go to the clinical studies of reentries. This slide is a summary of the work from Simon Ryan, one of his first summaries of his data. So he takes this basket catheter, 64 electrodes on 8 splines, and he puts that in the right and left atria. And here are the activation maps that he observes. He sees rotors, and he sees breakthroughs, focal activity, and he ablates them, this or that, and he gets, after a relatively brief ablation time, he gets transition from AF to sinus rhythm. And his data suggests that what he reports is that in 98 out of 101 cases in sustained AF, he saw focal and impulses and rotor impulses. There were about 2 sources per patient. 70% of those sources were rotors and 30% were focal. He reports also that there were more sources in persistent versus paroxysmal AF, and he suggests that those rotors and focal discharges were important for AF because when he ablated them, the AF was terminated acutely, not after cardioversion, but rather at the moment of ablation. We have some study and some simulations about that type of recordings or that modality. We suggest that there may be false positive results here, that some rotors that were reported as rotors may not indeed be rotors, and those focal activities may be also re-entry, so it's not so clear. However, this mechanistic role demonstrated by ablation seem to be very strong point in favor of those discrete sources as being important for driving atrial fibrillation. Now, another modality, that's the one that you are familiar with more than me, the ECGI is used in Bordeaux, and here you can see some of the data. I'm sure you're involved in generating those. Here you can see a rotor in one patient during AF, and this is another case with maybe more rotors and less stable. They seem to drift. And the group of Bordeaux summarized their 102 patients in persistent AF by saying that in most patients, drifting rotors were sustained for 1 to 7 rotations. And in terms of their mechanistic role, they concluded that pre-procedural mapping, delineation of critical regions, those that have sources, simplify ablation, particularly in AF that lasts less than 6 months. So the two modalities, the basket catheter and the ECGI, seem to find rotors. It's not so clear, not 100% clear what is that. I have to say re-entry, not rotors, because we don't know if they are functional or anatomical. But there seems to be still the questions that whether the modality is affecting the observation and how should we interpret those recordings and those the data processing. Now, what I want to show you, yes.
So are all functional reentry, are they all rotor-based reentries?
That's, yes, the same thing. When you say functional reentry, you say rotor, yes. It's the, 2 words for the same thing. Please check for us, and I suggest for everybody, because otherwise we don't know what to talk about. Okay, so we had our own study of atrial fibrillation, body surface mapping of atrial fibrillation. Our vest had 64 electrodes. Is arranged in this diagram. We looked at 14 patients during EP study. The patients were very heterogeneous. Some were paroxysmal, some were persistent, some had previous ablation procedures. So what I'm trying to say is that we cannot say anything about the AF here. What we're going to say now is mostly about the mechanisms of mapping rather than the AF, okay? Because I cannot tell you what happens to patients that just had AF yesterday or patients that had AF for a year, because the conditions didn't allow us to do that. Now, in terms of signal processing, we remove the QRST either numerically or by adenosine. When you apply adenosine, you have a node log, so you have a few seconds without ventricular activity that enabled us to to analyze only the atrial fibrillation activity. We look at signals that are typically 4 seconds long. We did FFT, invest dominant frequency, and we applied the Hilbert Transform for get phase movies. For validation, we use the Carto system. It's a sequential mapping, but it enables us to look at the dominant frequency from inside the heart with validation, looking at the power spectra and the time series together. So we could not just get a number that the system will give you. It was not just a black box that throws you numbers and then you have to either accept or accept. Anyway.
So just one quick question.
Sure.
Given the adenosine affect the properties of the atrial fibrillation?
It accelerates.
It should accelerate it, but does it change the characteristic?
Well, the highest dominant frequency seems to be in the same location once we apply adenosine. It doesn't move. So we use it to remove the QRST. Initially, we used it to make sure that the dominant frequency that we measure is not an artifact, because there could be several issues with dominant frequency, like harmonics, subharmonics. Sometimes you have ambiguity in which is dominant frequency. And if we're interested in the activation rate, applying adenosine, challenging the AF to accelerate it, we know that should be accelerated. That helped us to have another condition to confirm the value of the distribution of the DF. Where is the highest dominant frequency? Because we are not so much interested in the absolute value of the frequency. We're interested in gradients. So applying adenosine, accelerating certain regions would actually enhance those gradients and actually slow down sometimes this low frequency. So that helped us to locate the highest dominant frequency site. What I'm showing you here is a comparison between, well, here's a comparison between the inside and the outside. Here's what we got on the body surface. This is, well, it's not in units of frequency, it's the units of difference between the inside and the outside. And when the left atrium was the fastest, the difference was 0 in the center of the back. That's the center of the back. That's where the activity from the inside matches the activity on the body surface. When the right atrium was the fastest, is another patient, that match occurred here. This is the site that reflects right atrial fast activity. And if we look at 14 patients, when the left atrium was fastest, that area in the center of the back, a little bit on the left of the back, was where all the highest dominant frequency were located. And when the right atrium was the fastest, that high frequency region seems to concentrate on the here under your right arm. Okay, this is of course a region that typically you don't have any recordings there. Nobody is going to put a cordial in those regions, and we suggest it may be a good idea to start moving those electrodes to those regions. This is the graph that shows the correlation between the inside dominant frequency and the outside dominant frequency. On the left side, we see good correlation, and on the right side, inside, and the right outside, again, good correlation, not 100%, but good correlation. Importantly, the opposite side give us low correlation. For example, left versus right, here is the regression value is very low, and the right versus left also is very low, suggesting that we have, I wouldn't say 100%, but we have high sensitivity and specificity for the spectral properties from the body surface to what happens inside. Now, what happens in phase patterns? We start with atrial flutter. So we expected very simple activity during atrial flutter, and indeed we see these are phase snapshots at three different times. We see two counter-rotating random activity, and that's what is expected during flutter, because if you think about it, clinicians describe their flutter as re-entry around the valve. However, in the atria, you cannot have only one re-entry. There must be always, from topological principle, there must be always two re-entries. They always come in pairs. Okay?
I'm sorry.
Yes.
These are phase plots on the body surface,
On the body surface, yes.
not on the heart.
No, we don't do the inverse solution, sorry.
Right, so these are on the body surface, and it's a phase plot based on the ECG.
Based on
Of
the
your
64 recordings.
ECG
Yes,
record.
class, yes.
And you define phase based on the ECG signals. I mean, before you showed your phases and the action potential.
Yes.
Now it's not an action potential.
Exactly, yes.
It's the electrocardiogram.
Yes.
And you define phase there, and these are plots on the body surface.
Yes. Okay. So So if I go back to that, so we have two phase singularities, two singularity points here, counter-rotating, and that was expected in flutter because you must have two re-entries during flutter. Okay, one is around the valve, the other could be around the vein, superior vena cava, inferior vena cava, or around the crista terminalis, but there must be two, and that's what we see here on the surface. This is a panoramic mapping, so we must have two. Okay? So this is a simple case. What happens during AF? The situation is, of course, not as simple. Here I show you two different processing of the same data. So this is a phase map of unfiltered data. I would say minimally filtered. It's not that it's totally unfiltered. What we see is at different times, this is 140 milliseconds apart, the singularity point that was shown here, the center, jumped to this point, and what we found is that during AF, the singularity points appear on the surface, but they are moving erratically. There is very little we can say. They seem to move all over the place, and we were not able to interpret them. So we looked at different frequency bands. And then the situation is much simpler. And in particular, when we filtered around the highest dominant frequency that we see, either from inside or from outside, they correlate. Now we know that they correlate. And once we filtered in the highest dominant frequency, we saw the singularity points stabilize tremendously. They, I'm not saying 100% stable, but they moved much less. This is a movie of that case in which we have filtered that ECGs, those 64 ECGs, and then performed the phase analysis. Oops.
What kind of frequency constraint did you use there? What was the
That-
window in terms of the frequency
Plus
constraint?
minus 1 hertz. So it's
Okay.
a very narrow band.
Okay.
Yes, good question. Good point. So, so this is the singularity point that we see, and it seems to move a bit, but that's for us stable. That's for us it's stable because without filtering, we couldn't even track it. Now it disappears a little bit. Goes here. Remember that our vest reaches about this height, and of course, it's a good question. What happens below that? I wish there was some way to go and measure the ECGs on the diaphragm, but we cannot. So anyway, so we see sometimes one, sometimes two, sometimes more. Sometimes it's moving outside of the field of view. But that's the situation that we have. One of the examples that we have. Here's a summary, or no, sorry, another example, a little bit more details of a patient with unfiltered and filtered phase maps. The filtering is done before the phase map. So before we looked at 6 signals, 6 electrodes in this region, and before the filtering, those were the black traces. They were very complex ECG traces. Here are shown the 6. They all, nevertheless, had a same dominant frequency, 6.75. Once we filter that map with very narrow band around 6.75 hertz, they became like the blue traces, and we can see that they show continuous rental activity as illustrated by this very, very stable singularity point. Now, when we summarize all the patients, we saw that the percentage of time with rotors on the body surface went up from 7%, 8% to about 70%. Very similar to the percentage of time with rotors in the studies by Narayan. The duration of the rotor was jumped from about 50 milliseconds to about 400